Welcome everybody to today's deep seminar. It's a pleasure to have Professor Wilson Poon here. He's a professor of natural philosophy at the University of Edinburgh. And we're very excited to hear from him why we should work and study the physics of death. So please take it.
Right. Well, thanks for inviting me here. Amsterdam is one of my favorite cities, so there's always good to have an excuse to come back. So I've been thinking about death as a science question for about 20 years. When 20 years ago, someone invited me to give a talk on complexity and decay. I have no idea why they invited me to give that talk, but that was the stage I started asking, what's the difference between saying my computer died? I didn't mean just run out of battery. I mean just, I need a new computer. What's the difference between my computer died and my cat died? And I started asking myself, what is special about biological death? And I got more and more excited as I thought about it. So here's everything now. So it took 20 years to really get to the point where I had results to present. It took 15 years to really believe that this is an interesting question. It took 5 more years, with 3 years of COVID in between, to articulate research questions and to start collaboration to actually have some results to present. Now, can I just check the composition of the audience? Who self-identify as physicists? Just so I know. Physicists, so quite a lot of physicists. Biology, who self-identifies biology? Any biologists? No biologists in the world.
He's a biologist, some sort of biologist.
Yeah, we were waiting for you.
Just like, well, I need at least one to tell me that I'm talking rubbish. Any other subjects in the audience?
Mathematicians.
Mathematicians? Yeah, okay. More and more. Oh, okay. So it's predominantly physics. Okay. Anyway, I'm aiming this at an interdisciplinary order. So if there are things that are either too elementary or too difficult, just let me know. Okay, so there you go. So this talk is a slightly more technical version of the talk I'm giving in the Institute of Physics this afternoon. Thank you, Massey, for the wonderful poster. I've never given a talk with a poster like that before. Am I standing in anybody's way?
No.
So I have 2 parts. Basically, I want to, after introduction, setting the scene, which is quite a long introduction, to explain to you why I think this is worth doing. Part 1, I'm going to talk to you about how bacteria disassemble their dead bodies. And so that's quite a lot of biology. That's basically an experimental biology sort of result. We would have names of genes from it, 3-letter acronyms. The second part, I will set up some sort of Ising model for active cell disassembly. By Ising model, I mean this happens on a square lattice and has no units, and almost anything goes, but it has some relationship to reality if you think hard enough. So that's a sort of theoretical physics type result. So
And wave your hands enough.
Sorry?
And wave your hands enough.
I'm going to wave my hands.
Wave.
Right. So look, here's an interesting paper in PNA, not PNA, Open Biology in 2017. So it talks about tracing the dynamics of gene transcript or organismal death. What it's doing is that it looked at gene expression in the, I think, the liver and the heart of zebrafish and mice. Okay. Somewhere in this paper it says this: after death, an organism undergoes a thermodynamically different process of spontaneous disintegration. In other words, the picture that these authors, and they are distinguished people, I mean, Peter Noble, famous biologist from Oxford, systems biologist, their picture of death, or at least their picture of the afterlife, is a process where basically, if you like, the second law takes over. Okay, so finally, equilibrium wins. After death, the organism is no longer non-equilibrium, and so the second law takes over. The thing just disintegrates, maximize entropy. So the picture is that of a car left in a field to die. It just falls apart. Now, what they actually found was this. What they actually found was that, so this is what they said, in life, genetic and epigenetic networks precisely coordinate the expression of genes, but in death, it's not known if gene expression diminishes gradually or abruptly stops as specific genes are partly lost. So they looked at the post-mortem gene transcription. We don't need to know the details. These are different genes, I think, in zebrafish and in mice, same homologous genes in both. And what they find is that the pattern of gene expression, the number of hours post-mortem going up to 96 hours, 4 days, is highly specific and highly reproducible. In fact, the forensic scientists are very excited by this because someday in the future, you may be able to look at gene expression in the liver of a dead body and figure out precisely when they were killed. So it turns out this is reproducible. The pattern of gene expression is specific and reproducible.
But is it different from before that?
Well, I mean, yes, because before death, well, yes, but I mean, before death is the entirety of his lifetime. So it depends on the zebrafish is swimming or sleeping or.
Well,
I mean, I'll say yes, but that's almost a trivial answer.
well, no, I don't know whether it's a trivial answer. I mean, these things are driven by ATP in cells.
Yes, indeed.
So there is, on the moment of death, there is of course still an amount of ATP
It's a
available.
significant amount of ATP in the cell.
And so life processes will actually be supported on the basis of that
Yeah,
ATP
sure,
that's still there.
Sure.
So that's the background of my question, like the cellular processes will continue to basically in the same way as long as there is the energy to
Yes,
sustain them.
but it is specific to post-mortem. It is
So-
different from pre-mortem.
It's different from chemo.
I can,
Can we say that organic things is still alive even though the whole system is dead? Like if I'm a
I'm
human,
not
I'm
going to that question, that's a different sentence.
Mhm.
But there's no doubt it's that because they've taken the liver out, taken a piece of tissue out, mesh up the cells, so that definitely is the liver is taken out of the organ. Just shout.
Can we quickly follow up on this? So what is the definition of moment of death?
I have no idea.
It's your
And
questions.
I'm not going to answer that question because then I tell you to be irrelevant for me. I'm not the author of this paper. I'm only using this paper as a bridge. So if you ask me too many questions about this paper, we will never get anywhere. I'm happy to tell you what they actually found and discuss the paper in utter detail, but then you can read the rest of the sentence.
Can we ask what is your definition of moment?
I don't have one.
Okay.
That is one of those things I don't know how to define, but I know when I see it.
Does
I'm
it,
not going to get into that either.
you don't want any more, but still have one more question about this. Does it matter whether or not you take this piece of liver outside of a living organism or a dead organism? I mean, if you take a piece of liver out of a living mice, it still can't survive on its own.
It takes it out of, I can tell you what they did. I think they use, I don't think it was chloroform, but they use a gas to kill them something. And certainly the three referees in the paper agreed that the organisms were dead before they took the liver out.
So if you take a piece of liver out of a living mouse, does that give different pictures than if you take it out post-mortem?
I believe that is discussed in the paper, but I can't remember the answer is. I think it is different.
Apart from the fact if it's ethical or not to take a piece of liver out of a living mouse, but
That's yet another sentence. I mean, I'm happy to discuss this paper and turn it to the journal club, but then you're going to get to hear what I've done.
No, but we haven't read it.
It's your choice.
It's not your, it's just.
Then I move on from this paper. But the interesting thing about this is that they actually said, we all know that at death, the second law takes over. So what they said, partially in answer to your question, is that probably this is the same gene pattern of expression as when the organism undergoes severe trauma, because death is probably originally experienced as severe trauma. So this is probably a pattern of gene expression that the body goes into to deal with severe trauma, because you can't argue with the fact that death is a pretty severe trauma. So that's what they said. So in other words, what they seem to think is that evolution could not possibly have, in any sense, selected or adapted the post-mortem response of the organisms. It's unthinkable for this group, at least. So, in other words, in Stephen Jay Gould's famous terms, any post-mortem pattern of behavior must be a spandrel. For those who don't know what a spandrel is, so this is one of the sites in Rome that you must go and see next time in Rome, partly because there are no tourists there, partly because this church, Santa Sabina, was old when St. Augustine was born. This is one of the oldest still standing and functioning churches in Rome, and is off the tourist beaten tracks. It's a fantastic place. But that's a spandrel. That's the bit between two arches. So in other words, architects do not set out to build spandrels. But if you build arches, there will be spandrels. And so, of course, if you're an artist, you see a blank piece of space, you would decorate it. But the spandrels are not built to be painted on. But since you build arches, there will be spandrel, and if you show an artist, they will paint it. So Stephen Jay Gould called this a spandrel effect. So in other words, anything that is evolved, a function that is evolved for other purposes, but it happens to be useful, can be co-opted for something else. You see, not so any post-mortem function is a spandrel taking a ride on something that is a pre-mortem function, such as responding to trauma. They also happen to be, in some sense, useful for post-mortem. Is it slightly clear what I mean by spandrel effect? It's not law for it, but it's there, so you might as well just kind of use it. I mean, in Seville, there's so many Seville orange trees that the boys, mostly boys on the streets, are playing football with Seville oranges. But Seville oranges is not evolved to be football. It's a spandrel effect. It's not fun when they kick a spandrel Seville orange at you, because when they're overripe, they splat. But anyway. Right. So, I mean, you may know Benjamin Franklin says there's nothing as certain as life, as death and taxes. So, I mean, my wife is a tax lawyer, so I tell her that what this says is that living organisms do not do inheritance tax. Any appearance as if they are planning for the afterlife is a spandrel effect at best. Right,
There are exceptions to that.
sorry,
There are exceptions to that.
that mean what?
Oh, there are exceptions for spiders that actually where the body of the mother provides food for the offspring.
Oh, absolutely.
So, and there are exceptions. There are organisms that actually it's a kind of cannibalism where the body is actually used to feed offspring.
So the whole rest of the talk is to turn your sentence saying, in fact, there is no exception. All of biology is like that. All of that is like that. But theorem one for physicists doing biology is there's always an exception. But I'm going to try to show you that in this case there is no exception. All of that is like that. It's much more interesting than that. But I agree. Of course there are cases, but I'm going to show you that for molecules ecosystem there is no exceptions. All of that. So this is Eden Revenue presenting a tax bill to death. So I want to show you that in fact on all levels, from single molecules up to equal entire biosphere, living systems in some sense plan, meaning natural selection has in fact in some sense adapted death and function. So look, we all know about nutrient cycles. Okay, so the leaf tree dies, falls in the forest floor. So this stuff in forest ecology is called forest litter. Now, it turns out that you're going to tell me you're a forest ecologist.
No, I'm not.
No, you're not. So it turns out forest ecology is a Dutch thing. It turns out. I discovered this. But you don't have any forests. Oh, God. Where are the Dutch forests?
Well, the same way where the English forests.
This is gone.
They were used to build ships.
I understand. So I'm slightly surprised that forest ecology is sort of Dutch thing. But anyway, so a while ago, in 1994, in fact, so it was exactly 30 years ago, this guy from, is it Wageningen?
Is the Wageningen University.
Is from Wageningen, published this paper, which started the subject, litter decomposability and neglecting the bones of plant litters. And it continued, I mean, I think this is also a Dutch paper, I think Convert, he's somewhere in Netherlands too. But anyway, plant species trace. So the second paper says this: the decomposability of a species litter is consistently correlated with their species ecological strategy within different ecosystems. In other words, what they found, and the second one, so this is a theory paper. It's a theoretical analysis fitting to a theoretical model. This is an experimental paper when they show that tree species, the same species, but in different, or the same kind of trees in different ecosystems, tune how decomposable their leaves are according to the what microorganisms, what insects actually decompose it. Okay, it's not random. They don't let the leaf fall apart. They know the leaf has to recycle back into the ecosystem. So the ecosystem as a whole, so I'm going to make it clearer in the next paper. So this paper is about co-evolution. The trees, the forests co-evolve the decomposability. I mean, I don't know of a less cumbersome word, but decomposability is a difficult word to say. But the leaves, including the decomposability of the leaves, is co-evolved with the natural biome, if you like, or the organisms that actually has to decompose it. So in fact, so if you look at this sentence, I mean, I always thought it interesting that, you know, if a biology paper used the word afterlife and something going on here, you know, so you know, what they're saying is that is that plants somehow plan the afterlife as part of the ecosystem of the biome in the soil. So, in fact, nature has done one experiment to show you what happens if that goes wrong. It's a very big experiment.
So guys, it's related to the definition, I guess, but here it's not really plants death, right?
Well, the leaves, the cells of the leaves die.
Sure.
That is
So
death.
it's not the organism itself that dies. And does it
The
also?
cells die, and I'm going to show you that this happens on all limbed stems. So the death here is the death of the cells of the leaf. Yes, but nevertheless, it is.
So there are evidence that the rest of the organism in plants.
I mean, when the entire tree dies,
Yeah.
as I'm going to talk about, the same kind of thing happens. But it's not just the leaves; it happens in the tree trunk as well. And that's the big experiment that nature has done, because the origin. I mean, we have a lot of coal. This whole came from the Carboniferous period. Carboniferous being coal forming in Latin, and so you know, I mean, these are artist impressions, but evolution in the late Permian and early Carboniferous, evolution invented lignin, proper wood, you know, just real wood, and that stuff. Not just cellulose cell walls, but actual, you know, xylem making wood. Evolution invented wood, but no one told the mushrooms, because mushrooms are the only thing in the universe that can actually decompose wood. So it turns out that the fungus came a little late. They arrived on the scene a little late. So the wet weather, as well as the lack of fungus, the warm condition means that the whole thing dies. I mean, death, of course, is difficult for a tree because the wood, in some sense, is already dead. But anyway, when the whole tree unquestionably dies, those tree trunks are just lying about. They got to the bottom of the swamp and just lie there and became coal, because the mushrooms evolved a little late. Okay?
If they were earlier, they could eat the wood.
Yes, the mushrooms are the only thing that can eat wood, even today.
Well, termites.
Yes.
That was long before termites.
Yeah, it's not.
Yeah, but nowadays we.
All right, termites.
The fire?
So let me just add it to the slide. Let's just let's just do this. So what you're saying is what I should do is.
And these little insects that, you know, dig through your furniture and boktor in Dutch, but I don't know the English word for boktor.
You don't need the English word for it, but the key thing here is many years late. You would have long been dead before that, because what actually happened was that all these trees sequestered carbon, and nothing robbed the trees back. No mushrooms, no termites, no other insects whose English words I can't remember. They're not there. The trees sequestered the carbon dioxide, the earth cooled, and in fact, the whole thing nearly turned into global snowball stage. That's the Carboniferous extinction. Okay, so when that cycle of disassembly goes wrong, something very bad happens. We only just got away with it because the mushrooms came along, and very much later termites, the dangerous past by the time the termites came along. Okay, so that's how important it is to actually make sure that your system disassembles the right way to feed back to the system. So I don't think there is any exceptions. I think over the whole of biology, that disassembly bit is important. Okay, so the active self-disassembly of plants is needed to couple back to self-assembly. It's the active disassembly on the ecosystem level. Rotting the wood to feed back is the ecosystem spending energy, because the mushroom has to spend energy disassembling the wood. It's the ecosystem actively spending energy disassembling the wood to feed back into self-assembly for other bits of life.
Excuse me, you now jump to active. Where does the word active come from?
The word active comes from the fact that in order for a mushroom to actually disassemble the stuff, it is actually spending energy, ATP to do it, or GTP or other things. It's not the backward chemical reaction, which is equilibrium, which doesn't take energy to do. Chemical reactions in thermal equilibrium is reversible.
Yeah, but in that sense, but you know, I really oppose to this kind of this tight connection between life and biological processes and chemical reactions. I know that physicists and chemists are really inclined to do so, but that's not how biology works. And you also talk about natural selection. Natural selection happens through reproduction.
Yes.
So everything that happens after reproduction
Yes.
is not selected. It cannot be selected for.
I know you have that objection. I have data to show you that it does select.
Oh, I'm very curious about it.
So you should be. So you should be. I have been backed by evolutionary biologists for 2 years on this, and now I have data.
And you also talk about like then, you know, the selection at a level that goes beyond the individual.
Yes, I do.
At the level of an ecosystem, there is selection on a level of an ecosystem. That is something that has been discussed quite intensely in the 1970s, and you also quote Stephen Jay Gould, and he's the one that actually killed it. That whole discussion showed that it was flawed. And
As far as the discussion is still ongoing.
not among biologists and evolutionary biologists, I think, but
I can stop now.
No, I'm just curious. I will. I find it interesting. So go on.
Right. So, if I go on for a moment, what that story suggests is that plants can't afford to simply just fall apart in some random way. The way they fall apart seem to be evolved to co-evolved to do this kind of feedback thing. So, you know, the point about this lot, going back to that paper, of course, they look at the dead body on the lab bench. They don't look at the dead body in nature. Because in nature, when the zebrafish dies, it doesn't wait for someone to come and live around the gene expression. It gets recycled into the system. When a mouse dies, it gets recycled into the system. So, because they've forgotten that animals actually evolved to live and die in ecosystems and not on lab benches. So, you know, I mean, these are ecology papers, these are evolution papers. So, you know, I think we need to look with those keywords. So, in fact, my first serious interest in subject came when I discovered this paper. So it says how an organism dies affect the fitness of its neighbors. It's in American Naturalist, 2011. So how an organism dies is about whether they commit suicide or they're murdered. So in other words, is it apoptosis or necrosis? you like. So they took some single cell algae, some Rollus and Hardii, and they grew it up in what is known as defined medium, in which you mix the chemicals yourself. It's the minimum medium in which they can grow. You know all the nutrients in it, the chemical, you prepare a nutrient. Some of the algae, they trigger cell suicide, programmed cell death, that particular form of programmed cell death known as apoptosis. And the other algae, what they did was they sonicated. So just to use everyday language, the first is cell suicide, the second is cell murder, different forms of death. I think they did sonication because they want to kill the cells instantly rather than using heat to allow time to change the gene expression, and all the rest they just want to kill them instantly like that. I think the only reason why this paper didn't go to a slightly high impact journal was that they didn't actually go as far as looking for the molecular markers for that, to prove that they actually induce apoptosis. They use a well-known protocol for inducing apoptosis, so I assume they did it right. What they found was, suppose now you take the lysate, you take the death soup in each case, and grow some more algae in it. Okay, so the cells die, they lyse, they blow up. Use the soup to grow other cells in. The suicide soup grows better than in the defined medium. The murder soup inhibits the growth.
So what does sonication mean?
Ultrasound.
Ultrasound.
The ultrasound bath, the same sort of bath as you go to the optician to take your glasses into clean. Then you get.
Sonication imply, the result of sonication is that you completely destruct the cell in its entirety, and you.
So they didn't use heat because the cell heats up gradually and it could change the gene expression, respond to fun stuff, make toxins.
You blow it apart into pieces.
So these are cell explosion. You just go through.
Okay.
Right. So interestingly, what this suggests to them is that probably where this comes from is that it might have originated in some starvation response. So in fact, if the algae are starved, some of the cells might actually altruistically commit suicide to find nutrient for the other cells. And the reason why this may be inhibitive is fairly obvious because, in fact, intact chloroplasts are somewhat toxic to other plant cells because of the reactive oxygen species around it. So in fact, when a plant cell commits suicide, it carefully uses a lot of ATP to take apart the chloroplast into non-toxic components before it releases it. But if you blow up a plant cell, typically the ultrasound is not strong enough to blow up the chloroplast. The chloroplasts are released intact, the chloroplasts are not good for the other plant cells. So what is suggested to them, and there has been literature since, to try to build a tight evolutionary narrative to say how this kind of altruism could have evolved.
So you're suggesting such traits or evolutionary traits?
That's what they suggest, and they are apparently biologists. So, I can only take their word for it.
Like the biologist?
No, Rick Michaud is a well-known biologist.
So he thinks that this could have evolved.
So one question, how are the time scales involved? I mean, there's this sonification, this happens in seconds, or and this apoptosis happens in
Minutes.
Okay.
But it's not days.
So it's not in the case that, in this case of apoptosis, that just all these stuff in the cell had more time to decay using the second law of thermodynamics.
No, they don't decay. If I decided to make the talk so different this afternoon, but if you come to this afternoon, so I'll show you a movie of apoptosis. It's highly organized. The cell goes through a very well-defined stages process. And in fact, apoptosis is the Greek word for falling off. So because they showed the micrograms originally to a Greek professor, and he thought they looked like petals falling off a flower, so he suggested apoptosis. So it's a very well-known sequence of stages it goes through, highly organized, And in fact, the cell needs a lot of ATP to do it. It needs a lot of energy to do it. When the cell is knackered, it can't commit suicide. It can only in some pathetic way fall apart. So they want to do this. You trigger them into a pathway where they want to do this. Okay. Anyway, so that is in fact the paper that made me think, actually, this is interesting, and one might be able to do some research on this. So, you know, in all of these cases, it is a system, whether it's the whole ecosystem, like in the forest with the fungus, or in this case, the cell of the Trichomonas bernhardii, is actually expending energy to take themselves apart in organized ways. So, and the product of the disassembly, death in some generalized sense, is fed back into the system. So in this case, to make sure that what you feed back is something that is usable, not the chloroplast, which is in fact poisonous, but the components of the chloroplast. There's only particle physicists in the audience. I know there's an ex-particle physicist, but you know, I always say that that shows that life is not designed by particle physicists, because what they would insist is every time anything dies, you disassemble it back to quarks. Because that's the fundamental components of everything. The trouble about that is that every time anything dies, you have to start the evolution of origins of life experiment all over again. So, you know, there clearly is a level of disassembly at which it is somewhat optimal. You don't disassemble it back to quarks, you don't disassemble back to atoms, you don't even disassemble it back to, you know, H2, but you disassemble back to amino acids. So, but you must disassemble the chloroplast, for example. So, I know you don't like it, but I'm going to continue to call this active self-disassembly. I can also call it dissipative self-assembly, because any process that uses energy dissipates energy. So, you know, if I talk to an exclusive physics audience, they like the word dissipative more. So, in some sense, what I'm saying is that life is not, so, you know, here is where I agree with the biologists. A lot of soft metaphysicists of my kind says life is self-assembled soft matter. But whatever else you may criticize that statement, is missing out half of it. The other half is also self-disassembling, actively self-disassembling soft matter. That's what hit me. So let me now tell you my two stories. I don't have time to go through both of them, but I'm going to tell you a biology story and a theoretical physics story, and I think they do meet somewhere out there. So one of them is a story that I started with Martin Cand in Durham and Stuart West as a evolutionary biologist from Oxford. The second is a general physics type story that I've been working on with Carsten Kruse and Jean-Pierre Ekman. So let me tell you a story on organism level of what E. coli does to the dead bodies. So the best way to introduce this is through this thing called the GARS phenotype. So if you grow E. coli in a bucket with a fixed amount of carbon resources, glucose or whatever, so you don't add any more carbon or nitrogen sources in, the number of cells on a log scale against time typically goes like this. So this is in first year microbiology textbooks. There's a lag phase when they think about what to do. There's an exponential phase, it's a straight line. There's a stationary phase, and then the stationary phase crashes, and it can crash in two stages. These are big numbers. These are a number of years. But anyway, but what these people discovered is that somewhere in a week to 2 weeks, mutants start emerging. In other words, these cells are not what microbiologists call persisters. These are not just cells got into some dormant state and just hanging around. Because if mutants involve, if mutants are coming out, it must mean that they're replicating their genome and make mistakes and therefore have mutants. So these mutants are called GASP. And for once, a biological acronym actually makes very good sense. It says growth advantage in stationary phase. What it means is if you take these cells, these mutant cells, and compete them in growth experiments, number of cells against time, a low density of GASP, high density of the wild type cells from here, the GASP outgrow the wild type. They grow better. So that means mutants. I mean, they use sequence the genome. They know they are mutants. Okay. So what that means is that in this stationary phase, the cells are constantly doing the cycle. They grow, they die. The cells, the live cells absorb the death soup of the other cells, and then they go back. Okay, so they must be doing that. So in other words, these cells are not persistent. They're not just these cells that are hanging around at a very low metabolic rate, waiting for a better day to come. They're doing this. And it turns out that there's quite a big literature on this, but no one has ever seriously thought about this bit. That is my obsession. It turns out this bit is highly non-trivial in the following sense. So that bit is E. coli cannibalism, right? Cells die, the other cells eat the remains. Now everybody since Pasteur know that they can do that. And you lyse an E. coli cell, however you lyse it, whether it's cell suicide, which is what's happening here, or sonication, or thermal, whatever it is, everybody knows that when an E. coli cell lyses, other E. coli can use the nutrients. Well, you should always suspect what everybody thinks they always know. Because this is biology, there must be an exception, and the exception here comes from nasty physicists deleting just one gene. It turns out there's just precisely one gene you can delete that makes that process impossible. So here's the data. So they use mutants. So this is looking at the optical cell density, which they checked is proportional to the actual number of cells in the thing between 20 hours later and at the beginning. So this is basically how many cells have grown in the broth. So this is the wild type. This is the E. coli that you just put in, that they just got. This is this particular strain, which some of you may not recognize, but never mind. It's a strain that's used quite a lot in as cell factories to express proteins. But this is the wild type. So this particular mutant has two genes deleted, OMF-T and LON. These are both proteases. So what they do in the cell is they take apart proteins. These are proteases. You delete both of those, basically they grow less well than the cells in just pure water. Pure water, they grow a little bit because the cells can still lyse and the other cells can use the product to grow a little bit. But these really nothing happens. It turns out that if you take an E. coli, you just delete just this gene, just the long gene, but leave this gene alone, this is the same. But if you delete the OMF-T gene and leave the long gene, they grow. So in other words, the deletion, these cells don't grow because you deleted this gene. You deleted the long protease. Okay, so if you just delete the long protease, these cells cannot grow. So in other words,
So to the want to drop. Are there other functions identified for that gene?
yes, but I'm going to say a little bit more. They're both proteases. They're both in their pre-mortem function in the cell is to take apart proteins, to do protein treadmilling. Proteins are made, destroyed. I can give a talk on that too, but yes. So in other words, so let me be clear what they do here. These cells, you take the lysis, you take these cells, they die, you lyse them, and the question is, can other cells grow in their lytic product? Okay, so if we delete that one gene, it turns out that cells cannot use the lysis product, the death product, to grow. And that,
No, but you can also argue that you only show that just in general, its capability to grow is reduced. Also, if you would add some other carbon source, so you should actually compare it with, have a reference experiment where you do add a carbon source by hand.
there are 20 papers of control experiments in this matter.
Yeah. And they indeed show that if you add a other carbon source, that these
Yes, sure.
mutants can still grow normally as if they were.
Oh,
Okay, yeah.
yes, this is a biology paper, most of these control experiments.
Yeah.
Unlike a physics paper, which say no. No. This is all I's across, or I's, you don't cross I's, all I's are dotted, all T's across. Oh, yes. And many more, which we try to tell you. But so it turns out that if you want the cells to explode, die, lyse, and that its products are usable for other cells, there's a gene whose product, a protein, is needed. And that gene is long.
Can I press a little bit more on
Of
this?
course.
So why, so based on this paper and all the controlled experiments, so what's the explanation? So what happens mechanistically?
This is when I can use the satisfying answer is in the next slide. But actually, no, I think it's on this slide. I think it's here. There's a reason why there's a blank. So it turns out that when a cell explodes, dies, lyse, it releases the products. The proteins are released as whole proteins. And proteins are big. So lysosomes. Proteins are big. And E. coli cannot suck in proteins. It turns out that E. coli and other bacteria too, they can only take in amino acids, dipeptides, or tripeptides at most. It's not just the size of the whole because it's active transport, but broadly speaking, it's the size of the whole. They cannot take a whole protein in. Okay, so it releases proteins, which is what happens when the cell lies at the instant of lysis. It doesn't work. The other cells cannot do cannibalism. Something has to happen to the proteins. Now, of course, if you just, I mean, by the way, you may or may not find this interesting, but if you leave protein in distilled water at room temperature, sort of Dutch, Scottish room temperature, not Sahara room temperature, it takes about 4,000 years to fall apart, which is about exactly the same as you leave a polystyrene water bottle in distilled water. I mean, it's the same kind of bonds, carbon, oxygen, carbon, hydrogen. So yeah, I mean, if you leave the protein just there, fine. It will fall apart eventually, but by that stage, all the other coli are long gone. But it turns out that what this one gene product does, what the long gene product, which is the protein called long, is the protease, what it does is it chops up all the other proteins. Now, of course, it raises all kinds of questions. That means it has to be quite stable, or the rest of it. But nevertheless, what it does to answer Mazzy's question is the protease. We don't know why it has to be that protease, because it turns out that deleting the other protease doesn't make any difference. So it raises all kinds of other questions. But it turns out that it is a protease, and what it does is it chops up the dead soup into small, and we have done the metabolomics, we have done mass spec to check that there will be massive increase if we have the long there of dipeptides and tripeptides. This is metabolomics was done in Edinburgh. So what the long protein does post-mortem, it has pre-mortem functions too. Post-mortem, it chops up the protein. So that certainly blew the mind of Martin Kane. He is a biologist. He found this surprising that there's a gene with a post-mortem function. So the protease long has the essential post-mortem function. Sorry, this is the next slide. I told you I had data. Stuart West is a very good evolutionary biologist specializing in algorithm in bacteria. So it turns out that this is active self-assembly because it takes energy to produce the long proteins in the first place. Well, that's obvious. It needs ATP to build a protein. So the cells, the wild type cells and the long deleted cells grow at different rates. The cells that don't make the long protease grow a little faster because they don't have to spend the energy building the long proteins. We did the experiment, these are data. So long is, so this story is an instance of active cell disassembly. They have to put energy into produce these things later to do this disassembly. But the point is, this is then what evolutionary biologists may call a common good. So in other words, you produce this for the benefit of the community. So again, under nutrient limited conditions, some cells will commit suicide, and those cells that commit suicide, if they have the long proteins, if they have bothered to spend the energy to produce the long proteins in the first place, it enables the other cells to use this nutrient to carry on. This is a common good. As usual, the common good, you can subject to what is known as the tragedy of the commons. In other words, why not cheat? Why not produce it, but still get the benefit? So if you expect cheats.
But what what you do here is say that these energies, this extra energy that they invest to produce this long gene or this long,
There's one.
that it is tied to the post-mortem effect.
It's not just targeted.
Whereas
It has
you also said
pre-mortem
it has pre-mortem functions, or it does something pre-mortem.
It does.
And so,
Which is why I need the next bit. Just allow me the next bit and then you can ask the
okay,
question.
sorry.
Allow me the next bit. And the paper has carefully investigated its pre-mortem functions too. It turns out that the delta long is not as good as in its SOS response. Okay, so, but that's just between you and me, because they don't know what I'm talking about. But, yeah, so, you know, I mean, you have some significant pre-mortem function. It's not essential. Other genes can do it, but we checked and yes. Can't be done all the controls. Right, so it turns out, I mean, you need to read Stuart West's paper. Stuart West is a evolutionary, social evolution biologist and theorist. So it turns out, and I'm not going to go into details, you do social evolution theory, what you find is that in this sort of situations, the proportion of cheats goes down as the cheat density overall goes up. Turns out, I could talk longer on that, but it turns out, and we've done the experiment, and it follows the theoretical prediction. What happens if the cheat has been controlled through a natural selection process to a level that they don't randomly take over, but they're there. All the details are in the paper. And happy to discuss it later, but you know, you can write the social evolutionary theory down, and this is what happens. So in other words, it's not a standard. Natural selection has worked on it in the classic narrative of how altruistic traits can evolve and stop cheats. It fits that kind of evolutionary theory. That at least partially answers some of your questions. Not
I'm trying to understand the graph that you're actually presenting.
so this is the relative proportion of cheats.
Yeah,
Sorry, the relative thickness of the cheats
the relative, yeah.
versus either the total the cheat density, you know, the number of cheats per mil, or the total cell density. So one could do the social evolution theory of what happens if you have totally the common, but there's a feedback loop to control the cheats, and that's what happens. But when the overall cell density goes up, the relative thickness of the cheats goes down.
So what is this? What is this x-axis value? There's a proportion there. It's for the parentheses between -1 and 2. So I don't really get what you are.
I think that's a log scale,
It's a log scale.
which is why they're negative numbers.
Okay.
I did tell them they should put a log on, but they didn't listen to me. So that's how biologists tell it. Obviously, it isn't. But I did try to say it's a bit odd. Negative number. Well, I'm glad you raised it. I'm not going to tell them there's a biologist in the audience. Right. Okay, so I went through that slightly quickly, but we worked with evolutionary biology to show that at least he was convinced that this postmortem function is long, is not just expanding. There is some evidence that evolution might have worked on it to select it from that function. And so we checked it with bacillus as well, and it turns out it's the same with bacillus. So for physicists, if it works for 1 gram positive and 1 gram negative, then it's general. So at least it's not just colon. Bacillus cannot use the lysate either unless there's long in the lysate to cut the proteins up.
But excuse me, but did you also measure, because if the proportion of these delta long mutants increases, the amount of public good also decreases. So the conditions under which what you're showing there doesn't really hold, because you also plot the delta the difference in figures. If the public good is not there anymore, both of them will have the same things.
Can I send you the preprint?
Yeah, sure.
You read it,
Go.
I've got to develop tomorrow. Good. It's a little bit short even in the preprint because it referred you to Stuart West's other papers. Because he's done this sort of thing
I know
in
his
something.
work, so.
So this is repeating his work, but using this method.
Yeah.
Okay, right.
Can I ask one question?
Of course.
You mentioned that this long production is post-mortem, but how?
No, the
but
production is not
the
post-mortem.
production is in living
Yeah, sure.
cells, but it's for the common good of breaking down.
Oh, that's one of the functions. It really has a pre-mortem function too, as many pre-mortem functions.
But that's that seems to me a pre-mortem function because the living cells benefit of that.
It's a post-mortem function for the cell that has died.
Yeah.
It's a common good for the other cells that are still there.
Mhm.
So whether you call it a pre-mortem or post-mortem function, I'm prepared not to call it a post-mortem function if it doesn't help you. It's a function to enable when one cell dies, it has produced something that enables its afterlife to benefit other cells.
Oh, the dead cell produces the lung.
Oh, yes.
The lung.
Oh, yes.
Ah,
Oh, yes.
okay.
If I take cells that don't have lung, is dead cell doesn't produce lung pre-mortem, steps cannot be cannibalized by other
it's not
live
like
cells,
they're-
whatever, whether they produce it
The
or
other
not.
living cells can produce the lung to break down the delta lung.
No, because they don't secrete lung outside. There's lung remains inside the cell. The
It
only
has
way
to
to
be
get lung
inside.
outside is to explode that cell.
Nice.
No, that isn't both.
Then I go. Yeah.
Right. No, thank you for raising that because that is absolutely crucial. It is that way around.
Yeah.
So that's the biology story. I can stop now, or I can just tell you the theoretical physics story.
That was the physics.
Yeah.
Because, it will take me more than 6 minutes,
That's fine.
That's fine, yeah.
It's a relaxed seminar.
because I like not to go over time unless we give the explicit permission.
Oh, you have permission. You have permission to go over now.
Can you sign this? Right, so in particular, this is very dangerous, because basically, any seminar speaker will mention those words and just leave, because it's going to bullshit, but anyway. So I'm going to say a few things about. Sorry, I can go over time. It's recorded. Right. So this is a team from Groningen, I think. Does anyone know Sabran Otto? Is it Groningen? It's either Groningen or Wageningen, but I think it's Groningen. Anyway, so this, I generally don't like, where does it say that? Yes, I don't like journal editors saying this in papers, in journals, but in this case, I agree that this is a very important paper. So what I did was this. Now again, don't take me apart on their paper. I'm just going to tell you what they did.
Right.
And what they say about it. Okay? I'm not responsible for their paper. Right. So they wanted to do tested evolution experiments. People have done it with RNA for a long time. But Sabran is a good organic chemist. He doesn't want to use somebody else's molecule. So he made a molecule. He made a molecule that scares physicists, which is why in his paper he drew it the way physicists like. Because he actually draw the molecules to scare physicists, not read the paper. It's a beautiful paper because he has diagrams for physicists and diagrams for chemists. So he actually draw the molecules for the chemists to read, and then he draws his cartoon for the physicists to read, so we're not put off in the paper's very well. I will learn this in the future. This is a physicist version. So this thing has sort of arms and things, up to whatever it looks like. So these things can stack up and make polymers. Okay? So what he wanted to do is to impose selection pressure on his test tube to select out longer and longer and more, and so you call longer, more complex polymers. It's the minimal degree of complexity you can think of. It's not much, but it is that. So you want to do test tube evolution experiment, much like the RNA people. And it turns out that all his attempts failed because there's a well-known problem, which in fact turns out the RNA people knew about. If you don't do anything and just try to do your experiment, the dimers and trimers always win because they always replicate quicker. So he just ended up with some pathetic tube of dimers and trimers. Doesn't matter what selection pressure he applies, he ended up with dimers and trimers. And that's what the early RNA evolution experiments do. They end up with small RNAs. They never grow more and more complex. So what he had discovered, and I don't know how he discovered it, because I kept meaning to go to Groningen to see him to ask him. is that what he has to do I say this? Yes. He puts in a molecule, which he calls a highly reducing molecule. For physicists, that means a molecule with a very high energy electron pair. In other words, he supplies energy to selectively disassemble the dimers and trimers. Then it works. Then he can apply selection pressure and it goes up. So in other words, he has to very selectively by hand, this is not natural selection, this is human selection, but by hand, selectively actively disassemble parts of it. In other words, he drives some backwards reactions way off equilibrium. Then it works. I was very happy.
But if you destroy the small components, how can you build large components?
You destroy the small component into monomers again, and that becomes substrate if you have to build the bigger ones.
But then some sort of you need to then step over, so.
Yeah, but it doesn't
step
destroy
over the top.
all of it. You have to do the detailed chemical kinetics, and how much of it destroys.
So there should be enough of the two and the
Of
diamond
course.
target left, so that some can escape to
So
higher.
if you're a
Yeah.
theoretical physicist, the substantial materials make you very happy because there are umpteen number of couple of ODEs to do all this in utter detail.
Nice,
And it
yeah.
fits. Okay, you like a couple of ODEs, read the supplementary material.
Yes.
So yes, of course, you can't destroy them all.
Hmm.
There is a bottleneck you have to go through, but it works. But broadly speaking, the narrative is until you very selectively do some destruction of a very specific kind, so in other words, it selectively destroys faster the monomers and trimers than the long ones, then it works. So I was very happy. So in fact, what happened was we both spoke in an online conference. I gave the talk that I'm going to give this afternoon. And then he gave his talk, and he didn't cite my talk, so I was very angry. And it turns out that he actually didn't listen to my talk because he was assumed that he. So, but I was so happy because basically that's what it is.
I don't understand why if now we also check with the diamonds and trimers, why then they don't start aggregating?
Why don't they?
Why can't you wait until you've got only trimers and then they will grow into bigger structures? Or can you only have like single thing?
For that, you need the appendix with the couple of ODEs. It's in the details. The core strain explanation of this is that they always come in faster. They always assemble faster than the longer ones. So you have to get rid of that advantage somehow.
Okay, but so you can only get single ones to attach to the thing. You can't get two.
I can't remember how important it is for big multimers to join together. I can't remember. That is in one of the ODEs. I cannot remember whether that is a significant amount or not.
So it's a stability problem, basically.
It is a stability problem amongst other things. But yes, I have to confess I did not work through the entire ODE model.
But if the system of ODE that one gives you, if there are different time scales, is either a stability problem or just is it like a predator system? Or?
I haven't thought through how to answer that question. But I understand why you asked it, but I haven't thought about it long enough to answer that question, whether it really is one or the other. Right, it should be ASD. So again, active cell disassembly was needed. Possibly, what this shows is active cell disassembly of this kind may be needed even in the prebiotic soup. Okay, so you don't have to wait till cells. It turns out if you want to kickstart cell assembly to make life, you might actually need to have active cell disassembly of some kind, even from the beginning. So I mean, I'll tell you it's an ideal model, so this is it. It's a square grid, must be an ideal. Right. So what we did, what Carsten Cruz in the EPFL invented this model a week after he heard me give a talk. So what it was, so this is a square grid, n times n. Typically, I'll show you data for simulation data for 256 times 256. So we have these angles in it. So, you know, each one of these can have angles here or here or here or here. So these we call angles. So these angles can join together to make chains or squares. Okay, so the target is to assemble squares. I mean, I just declare this, you know, I have some target. So what I want is to assemble squares. So you do a Monte Carlo simulation, the sites can change one by one. You go for those who know, Glauber dynamics. Okay, so sometimes it will resemble squares, sometimes it won't. And so we give them some energies. So these, I will call, if they point the same way, I call this a cis joint. It has some level of stability. This is a trans joint. It has some level of stability. So I want to favor these. Okay? I want to favor them. How would you favor those? If you only can do equilibrium thermodynamics, so you accept the change in the usual Monte Carlo kind of way, you make a random move, you see whether the new state is lower energy than the state you're starting off with, and if it does, then you accept it. If not, then you accept with some Boltzmann weighting. So all of this, in physics terms, satisfy detailed balance because it's an equilibrium simulation. So what happens is this: you can ask what's the number of loops you can form. So keep saying we should plot this as fraction, but this is the maximum. Okay, this is the maximum number of loops in my system you can form. So this is the energy of the trans joint, the wrong kind, they give you chains. Negative JT is the trans joint energy. This is the energy of the cis joint, those ones that can potentially make my loop. And it won't surprise you to see that there are very few loops if you favor the wrong kind of joints. You favor the trans joints, they make chains. If you massively favor the cis joints, then you maximize the number of loops. That's how you do it in equilibrium thermodynamics. You just make the cis joint energy significantly bigger than the trans joint energy, the Boltzmann factor, equilibrium thermodynamics, you do the job. Is that clear? At least clear enough. And I can say all kinds of things about it, but that's basically what it is. So from that point of view, the model is kind of fun as an exercise for master's students, but you know, fine. And you can do a mean field analysis. When I say you, I mean Carsten and Jean-Pierre can do a mean field analysis analytically, and you can predict the number of loops. As you increase the cis joint energy, it goes up, and the mean field and the simulations agree at low bond energy. So, you know, we understand the equilibrium model. If you do the stupidest thing and say, how do I favor loops? What you do is you make the cis joint energy significantly lower. The trans joint, I mean, there's no brain. You know, it just proved we got it right. Now what you do is you introduce equilibrium self-disassembly. So we spent a long time arguing what we should call these things. I think version 1 says eaters. I didn't like eaters, so eventually we settled on splitters. Okay, so after every Monte Carlo step, when you randomly do your equilibrium swap, you have a splitter, and you just throw it at random at a lattice, and whatever it lands on, so suppose it lands on this thing, then it randomly changes each of these squares into one of the four. Okay, so each of these configurations can end up in one of the four with probability 1/4. That's all we do. And it's instantaneous. So we haven't done dynamics. It's unrealistic. Nevertheless, we want the simplest conceivable thing we can do. And we still don't understand that fully. Yeah, so okay, so do you understand what a splitter does? You throw it in, and whatever it lands on, it looks at the thing. If it is not a square, if it has dangling ends, then it just splits it. And of course, there is a chance that it doesn't do anything because it can end up with the same configuration, but that's very low probability. You will just randomly swap the thing. So typically, it splits a chain into smaller bits. And that's a non-equilibrium step because that breaks, in physics terms, it breaks detailed balance and it goes one way. No reverse.
It doesn't do anything when it lands on a square.
Correct.
Okay.
So if jumping ahead, you might think this is a version of what biologists might call proofreading. You can discriminate the correct thing from the incorrect thing. I don't want to press that, but yes, I'll just declare that is the case. Right? So this breaks detailed balance is non-equilibrium, which means that in practice, if you want to implement this, anything that's non-equilibrium likely would involve energy dissipation. Okay? We don't do the energetics in this model. 3 papers time, we might. We might say that the amount of energy you need to split it is proportional to the chain length or something. So we haven't done energy in our model, we haven't done time in our model, but never mind, this is what splitters do. So this is an active non-equilibrium or active cell disassembly. So this is that color map now. Okay, so this is the color map before. So this is the wrong kind of joints. These are the trans joints. These are the right kind of joints. These are the cis joints. So previously, the only way to get a lot of loops is by increasing the cis joint energy. Or the lower cis joint energy, because it's minus that number. This is the map. So to see what's happening, you basically have to take the ratio of the two to see when the splitters help. So let me take the ratio for you. If you take the ratio of the two color maps in your head, doesn't help. It turns out that, well, okay. So it's just visually what happened. So each color bit in here is a loop. You can't quite see it. This is a zoom in, but doesn't help because the zoom is not enough. But each color dot is a loop. You can't see the little loop I'm talking about. So this is same conditions without splitters and with splitters. I mean, it's everything by eye under these conditions. I mean, it's a no-brainer. Of course, there are conditions under which you put splitters and you get more loops. That's not the surprise of surprises. Under what conditions the splitters have? So this is what we do. So it turns out we play around when we say we are constantly saying those two guys in EPS mostly. What they did was they defined this ratio. It's a little bit complicated, but it turns out that if I just simply plot the ratio of the non-equilibrium case with splitters to the equilibrium case without, the data is a little bit noisier and it's not as pretty, so we define this thing. This thing goes from -2 to 2. Okay, you can work this out, but this basically depends on the ratio, the raw ratio of the number of loops with the splitters divided by the number of loops without splitters. Okay, but this thing goes from -2 to 2. But positive means that having splitters enhance what you can do with just equilibrium thermodynamics. In other words, positive G is when having splitters, you can do better than equilibrium self-assembly. But another way, positive G means that having active cell disassembly does better than just equilibrium self-assembly. Active cell disassembly favors. The surprise is here. I don't really want to see it, but so this is the cis joint, trans joint energy plane. So on here, this yellow line is when JT equal to JC. Okay. Now, it's no brainer that when JT is bigger than JC, in other words, you favor the wrong kind. When equilibrium thermodynamics favor the wrong kind of joints, that putting splitters in helps. And that you didn't, I mean, wrong.
Well, let me confuse. There's no one type of splitter that you can design, right?
So this is a single splitter. It just lands at something at random. If it is an open-ended chain, it just splits it.
There's only one way to do it.
No, there are infinitely many. Well, many of them, because it can split. So each side just takes random, each one of the 4 possibilities.
Okay.
So I will show you there's one situation in which a splitter can instantly give a square. In all the other cases, what it does is it produces more monomers to further down the line. Monte Carlo can make loops for you. There was another, was it you were going to be
If I understand correctly, so it can be only positive for the first few steps, right? In the process.
don't understand.
No, as you're looping, as the number of loops is increasing, it gets more negative. I mean, this is what you read.
This is not time, this is steady state.
Yeah, okay, all right, but okay, well, how can I see that? If I see that, I know you cannot see that in the dynamic process.
We can check the dynamics. I'm not going to show you, but because I'm already over not real.
All right.
So I don't discriminate between the time it takes to split a long chain and to split a short chain. I say it's completely
How is this different from just favoring loops more?
I'll show you. I'll show you. That is of the absolute essence of the story. However, it certainly surprised us. It'd be interesting to know whether it surprised you. Well, I'm giving it away because I told you it surprised me, but never mind. There's a little slitter here. This little, so let me just do this little bit is when cis is stronger than, cis is more favored than trans. You know, thermodynamics already gives you more loops. Nevertheless, it's blue. Nevertheless, the gain is positive. So that's the interesting bit. It wasn't bad. Was it angry?
I think someone forgot something in that way.
Oh, I see. This is altruism. It has evolved. Right, so it's group selection. Right. So what was it? Oh, yes. So there's a little sliver here where, although thermodynamics already favors the loops, splitting still helps.
Yes, and then it's still the same as just favoring loops a little more.
Is not, because if it is, you expect these to be lines radiating from the origin. In other words, you just rescaling temperature. You expect it to be just dependent on JC over JT. It is, this line is a curve. That's the interesting bit. Yeah, well, that's absolutely the point. Thank you for asking the question. That's absolutely the point. It's not just like rescaling in temperature and favoring one more, because otherwise you'll get radiating lines that, you know, but it isn't. So we thought we got it wrong, and we did it again. This is right. It really does do that.
Is it like adding a penalty for having a chain?
No, it isn't, because it doesn't just rescale your energies. If you just rescale your energy, the data should not like this.
Well, do you want to mimic the program cell death by?
I don't want to mimic anything. I want to ask utterly general theoretical physics question. Under what condition would building in non-equilibrium cells and disassembly would make a difference? I want to play with the simplest model that has it and ask, does it have any surprise the conditions under which it may have a difference? I don't want to mimic anything yet. I will say something about whether there is any biological implications at the end.
But there are cases active system to destroy self-assembly, even active follow it. I can discuss with you.
Yes, sure. No. I mean, this is supposed to generate exactly that sort of discussion. Exactly so. So the easiest way to see that this is funny is to go to 0, 0. So in other words, when the energy for both types of joints is just 0. Okay. Nevertheless, the gain is positive. So can we understand that? You can understand it when we understand it's a little sliver because it's just in the neighborhood of the origin. Okay. So I think what's going on is this. So let's take the case when you don't favor either, that there is no bond energy whatsoever. So thermodynamics doesn't favor either. So in equilibrium, there's a very small number of loops. Nevertheless, there will be a few loops hanging about. So now you ask, what happens to the loop population when you add splitters in at the origin? The splitters can't act on those because I declare it to be so, because splitters don't split the correct thing. However, if a splitter lands on this, there's a 1/4 to the power of 4, which is small but finite probability that it leaves these three unchanged and it just turns this one this way and it instantly makes a loop. Okay? It's something like that. And if it acts on this, it would disintegrate into monomers, mostly, that can form substrates, single corners, to make loops, potentially make loops in future Monte Carlo steps. Either way, it helps to make more loops. But that's why at the origin, even at the origin, the thermodynamics doesn't favor either. Putting the splitters in already enhances it.
So you said you were surprised, but me sitting here, it sounds like the logical thing to happen.
That's exactly what I thought in the first part of special relativity. I thought how true it is that you could just explain it to a 6-year school student. Didn't stop Einstein being famous. Understanding it is theoretic. Getting there is highly non-trivial. For some of the best discoveries in the world, looking back, you wonder why no one has seen it before. I've just explained it to you. So, why
are you surprised that if you put in a filter that favors the thing you want, you get more of the thing you want?
No, I'm not surprised that can happen. I'm surprised it happens even at the origin, or there's a spinning phase space where thermodynamics already favors it. So I want to understand under what conditions, when putting a splitter, would enhance the thermodynamics. Because you see, up here it doesn't. Up here, by the time I get up here, so this is the JC to the JT line. Up here, when thermodynamics already enhances it, putting splitters in actually doesn't help. The effect is negative.
Yeah, so the surprising part is more that it is negative there than it is positive.
It's all right in the manuscript. If you referee it, make sure you read to the end, because in the manuscript we say both are surprisingly explain both. I choose only to explain one of them to you, but I can explain the other one to you too. But yes, I mean, the fact that this is non-monotonic, the changes like that, and there's a little sliver here, both are features that we need to explain.
Yeah.
I agree that once I've thought about it, I thought actually that is quite interesting, because it perhaps shouldn't have surprised me, but it did surprise me, and I talked to a number of other people and they surprised them. And so I had only 3 initial data points. The 3 of us involved in the project is surprised all 3 of us in the beginning. So,
yeah.
I mean, surprise is subjective.
Yeah, sure. Yeah.
Clever people tend to be surprised by less things. So, I mean,
or it's just the presentation, it's also clear. Yeah.
well, interestingly, one of my most cited papers is in fact a theory paper, so that's a nasty rumor out there around the theories. But I did this theory that afterwards I thought was utterly trivial. And my senior collaborator at that point, Peter Pusey, said, well, actually, it's only trivial, you because you understood it.
Yeah.
He thought actually it's highly non-trivial. So occasionally, for the discovery, when they come true, we understood it. But anyway, so let's not go to that philosophical one. But fine. Now, the question is this interesting or not? And I think it might be slightly interesting, because look, if I want to favor the loops, the most obvious thing to do is to just decrease JC, make the cis joins stronger. That's the obvious way to do it for those of us who are trained in doing thermodynamics. You see, biology doesn't like strong bonds. Of course, you need covalent bonds to bind the fundamental macromolecules, but macromolecules together need weak bonds to work. Now, of course, weak bonds are rubbish for specificity, for selectivity. The way biology solves it is to use multiple weak bonds. It doesn't do one strong bond, it uses multiple weak bonds to achieve selectivity. Because you need the weak bonds fundamentally for the thing to function. So I think it is actually interesting that it is in the weakest possible bond limit and in this vicinity that actually active self-disassembly has an advantage over equilibrium. So one could imagine that in very early even prebiotic conditions, when bonds are weak, without having to make bonds too strong, you can increase selectivity, you can increase the yield of the product that you want using weak bonds, but put in active self-disassembly. I'm sticking my neck right out, but it is interesting, is in that limit, that for me, the surprise happened over here, not over here. I'm clutching at straws, but that's it. I think that's all I have to say. So I'm trying to say that biological death is not just things falling apart. It involves highly organized active cell disassembly, which therefore enables particular optimized feedback. Biological death from that point of view is an emergent phenomenon. So I'm suggesting that there is enough generic level questions about the physics of it, but perhaps one ought to try to set one such physics up. And I think that, so the implication is not just for biology, but if you can do synthetic materials, as Sarah just said, because you can use it to make colloidal structures, but also to think about a circular economy because the principles are the same, and that's all I've got to say.
We have already a lot of questions, but we can take a few more questions.
Take them over lunch.
Yeah.
Also
possible.
No, I have one question. I have a remark and a question.
go ahead.
So what you show is surprisingly close to my own research. In which there
It
appeared,
must be good.
there, no, there appeared, no,
No, it's not something else.
so there appeared an article in the New Scientist 2 years ago about my work that's called Life from Death.
I might just have seen it briefly, but if I haven't, I should go and look it up.
So I can completely understand why you get all these results. I mean, the basis, one of the central results in my research is that actually when you apply additional mortality on a population, you can actually increase the biomass of the population. It helps to kill off.
Yes, in fact, I have read enough.
Yes.
Now, depending what it's about, I have read.
So, but what you don't show is that it's selected for.
I haven't shown it definitively yet, but I gave you some.
Because that's you don't, especially in the model, you
No.
in the model you show that like what I would call mortality, like destructive things, has an advantage because has a positive effect because you basically relax competition. It's my terminology. You have resources, which are these blocks.
I understand what you said, but I don't think we can say it there yet, but yes.
And but that it would be selected for is something that I've looked into, and you
can still select on like a population still, right? If you've got like two populations, like one forest where the leaves decompose faster, then that forest will grow enough to take the other one, right?
Yeah, but the problem is in the cheaters.
Yes.
The problem is in the cheaters. If you have a forest where trees produce leaves that decompose faster or whatever, then if there is a mutant in that forest that doesn't produce that particular type of leaves, doesn't pay these costs, but just gains the benefits of it.
That's assuming
It takes
costs
over.
something to make your leaves decompose
Well,
faster.
so this is, okay, so if there are no costs to, if there are no costs to a particular function, that's what is called the Darwinian devil. Basically, you can do everything for free.
Which you have to have a comment, right?
Not necessarily. No, this is about evolution. So if a mutant arises that can do everything optimally and it doesn't bear any cost, then that will take over, of course.
I guess you can also look at it from this like pillar analogy, right? That there is proteins that decompose things because you need them anyway, and that still work after that.
Yeah, but that's a side effect. And you have to show that it's actually selected for this. And selection at higher levels of organization than the individual is a very difficult issue that there is no answer to. Selection, for example, selection, because this falls all under the broad umbrella of selection for altruism. Why is there actually, behavior or processes that has a negative effect on the individual, but has a positive effect on its neighbors or its fellow individuals?
But there aren't these endless ideas in game theory where you have this sort of idea that if 95% of the population does something for the common good and you have 5%, then that isn't so egoistic thing, then this is a stable equilibrium kind of idea. That if 20% does the egoistic thing, then population collapse. But if you have this small fraction, then I mean, there are many examples in populations where you always have this small group of cheaters.
Yes, but it's, the problem is that this requires very specific interactive structures. So for example, that you, this is, they always have to be then like iterative games. In game theory, it's always iterative games that you play many times against the same partner or something like that. And so you need very specific assumptions to actually explain this evolution of unselfish behavior, let's say, or altruistic behavior.
I mean, I agree that the area is an active area of research because altruistic behavior, such as you running out with someone's phone, is really there. So the question is, it was purely a standard or not?
Well, I mean, so why we do this is like, is one of the biggest questions that we cannot address. And I also think that it's one of the, so cooperation in general is just a very big evolutionary question.
But is it clear that altruism is actually a thing?
Sorry?
Is it clear really that altruism is a thing and is evolutionary selected?
Well, no,
Is it true?
Well, that's a separate question, right?
it's whether so that there is cooperation. Let's call it altruism and cooperation. I use them interchangeably. That there is cooperation and altruism, that's clear.
That's clear. Okay.
That's clear. It's like it's absolutely clear that not only humans but also other animals actually have altruistic or display an altruistic behavior. But how it evolves is something that we don't know. How it has evolved. What is the
But altruism as something that also, I guess in your definition of cooperation, that has some benefits for the individual too. So it's not death for something else or someone else.
so
So
one
there's
of
the
the
individual benefits too.
so one of the explanations for altruistic behavior is what they call inclusive fitness. And so there is a famous story that so there are bird species where actually siblings help their parents raising raising a new nest. So this is altruistic behavior that was considered useless and considered as non-functional, non-essential, and so on. And they compared it even with things like homosexuality, that was also aberrant behavior, until 1966, when Hamilton actually came up with the idea that, who theoretically showed that this could actually be selected for if you had a relationship, a genetic relationship with the person that you helped. Because then you have a kind of then you part me and my siblings share genes. So to promote the gene, it actually you can come up with a theoretical argument that fitness is not just the fitness of an individual, but is actually the fitness of a clan.
I didn't know. That's right.
Maybe if I want to relate back to the talk or with the example of the bacteria that have that gene knocked out that promotes the decomposition of these long proteins. I think there it's even like almost necessary that the gene is there, otherwise the sort of the death soup, as you call it, is not able to sustain.
But there's also where Hamilton's rule by applies most obviously because they are, very highly related.
That could be, but so in that in the example you showed, for me it was not clear that it's clear that that this protease decomposes the soup and hence provides resources for the cell, for the other cells to grow. That's a kind of, it could be a side product of its function during life.
You could be, yes, I agree with that.
And that is there. There are of course many things that, or there are more things in life that, you know, are not really selected for, but that actually are just like side products of it. Yeah.
Well, I believe Stuart West's narrative is that that kind of result that we show the density dependence of the fraction of tutors suggests that it isn't. So I need to go and understand that much more deeply than I currently do at the moment, but that's certainly his narrative.
Yeah, I'm curious about that plot.
So let me say you like, you know, there I'm 95% relying on Stuart.
Yes.
I'm not understanding fully yet because he referred back to two of his own papers, which I have read. I can't say I fully understood the one I own it yet. But I mean, that is what he's saying is entirely clear too. So you know, at least if anybody says this may well be true, then I have to at some point take him on just. But that's why we got him to do those experiments because it could easily be a standard effect.
Yes.
Yeah, clearly it could be. But apparently, perhaps it isn't. Which is worth further investigation, I think, because it's quite a simple system to play with.
That's absolutely Tom.
And it's even ethical to kill bacteria, so no question about it.
All right, I think it's about time to go for lunch downstairs, continue discussion there, but first let's thank the speaker.